So that is important. And hatching. So which season? Okay. So how many eggs they lay? All these are very important. Then fingerlings. So what is the adult stage? Okay. All these are necessary. So this is about fish. Then prawns, they have larval forms which are called as nauplius. Nauplius. Okay. So they again develop in the next stages are zeomyces, post-larval stage, and finally they mature into adults. So first one is life history of rohu. So after its birth, so only after the second year of life, it attains its maturity. That means adult form. And it spawns only once in a year. That means it releases its eggs only once in a year, and that too for a very short period of time. Okay, and this season it coincides with the southwest monsoon. So during southwest monsoon season, it usually releases its eggs. So once the eggs are released, then fertilization takes place. And the fertilized egg is round in shape. It is transparent. It is non-adhesive. They won't attach to each other. And they are red in color. And within the egg, we all know yolk will be present. So yolk sphere contains no oil globule. Okay, there will be no oil globule here. And fully fertilized eggs are, the diameter of the fully fertilized egg is around 5 millimeters. Whereas once the egg is fertilized, then it will enter into larval stages. So the newly hatched larva, it measures around 3.5 to 4.5 millimeters in length. Again, it is transparent. It is laterally compressed body, and it has gill slits. So gills for respiration. Okay. So gill slits are present at that stage itself. Then pectoral fin is present, and median fin fold are present. So these are the characteristics of the larval phase. Then the yolk is gradually tapering. So it will be fading usually with a prominent anterior bulbous portion and elongated, and it forms narrow posterior blunt end. So which will be having slightly pinkish color at the dorsal end. Okay. So it has eyes, fish brown eyes and have slight pinkish hue at the dorsal part. And this hatching is irregular movement. Okay. So it's not a regular movement. And this spawn, just after absorption of yolk, okay, they releases their eggs. And the rudiments of dorsal and the pelvic fins appear on the 30th day, and barbels appear on the 21st day, and liver appears on the day of hatching. And the air bladder originates around 2.5 days after hatching. So this is the time period of development of different organs or appearance of different organs. So once this is done, so what are the characteristics or what are the growth stages in the rohu after hatching? So after 24 hours of hatching, so usually the larva will be around 5.5 millimeters in size, with a few black chromatophores on the head above the eye. And the embryo is pale yellow in color. Anus, which is the organ for excretion, so that is distinct, that will be present. Then auditory corrections, clear, auditory for the hearing. Then yolk sac ends sharply, distally to a point, and striation, that means like strips, are seen. Then pectorals are without rays. Then notochord. Notochord usually develops into the nervous system in future. So this notochord is turned upward at one end. Then after 36 hours, so the average size will be increased. So it is about 5.9. So previously it was 5.5. Now 5.9 millimeter. Lower lip is clear. Pectoral fin is prominent. Then anterior profile of yolk sac is also straight. Okay. Then again same black chromatophores are present throughout the length. And dorsal and ventral sides of the embryo are again pale yellow in color, then deep yellow, which is present in the caudal region. And the notochord is slightly upward at the tip. And here mouth appears as a slit. So we will see all these stages in the figure. Okay. The next is after 48 hours of hatching, the size has increased. Now the size is 6.2 millimeter and the yolk sac is convex. So it's lightly protruded outside. Then embryo is yellow in color. Air bladder is present. Pectoral fin is prominent. Black chromatophores are present, which are present behind the eyes. And no chromatophores are present on the ventral side of the air bladder. And auditory corrections are up. Okay. Then large black chromatophores are present on the head, which are prominent, which prominently appear. And fins, ventral embryonic fins appear. Okay. Head dark and body is there in the faint color. Okay. Faint yellow color. Whereas head is in the dark yellow color. Then after 72 hours of hatching, now the average size is 7 millimeters and looks pale yellow in color. So black chromatophores are present on the head, few are present between the eyes, and the embryo is bright yellow in color till the region. The caudal region is above the notochord, which is yellow in color. So I will zoom this, you can see. So how the embryo before hatching, but just after hatching and after 6 hours, so it is getting elongated and the notochords are present there. Okay. So you have fins, gills, everything appear one by one. Okay. This is till by 72 hours. Usually we can see mouth. So you can see that black spots, black chromatophores, they have increased like at the head portion. Then you have notochord, all these are present, which are elongated. Okay. Now next, this is about 4th day to 25th day that we will see in the, yeah. Next, the developmental stages of Rohu after 3 days of hatching. So that is first 3 days. So how the development occurs. This is after 3 days. Okay. So stretching from the abdominal region up to the anus. So anus is the excretory portion. So excretory organ. From that it is stretching. Okay. And dorsal embryonic fin is visible as forward to the opposite to the anus. So in the opposite end of the anus. Then 2 black chromatophore clusters are present at the caudal peduncle and 2 crescent-shaped patches are present in the caudal fins. Then caudal region is having 22 branched rays and it is less deeply forked. Then pelvic fin with 2 to 3 rays and dorsal with 2 to 11 rays. Okay. Then next is 4th day after hatching. So it is the size is increased. So your yolk is completely absorbed. So because you see in the egg, yolk is prominent, whereas when it matures into adult stage, so the yolk will disappear. So that yolk, why it is important during egg phase? That is because it provides the nutrition to the egg. It contains all the nutrients. Okay, but whereas after it becomes adult, it can feed. Once it starts feeding, there is no need for the supply of nutrition from yolk. So therefore, so after it forms the adult stage, usually the yolk will disappear. So yolk is completely absorbed on the 4th day after hatching. Lip margins are fimbriated. Then this distinct black chromatophores are seen behind the eyes in the head region. Opercular outline is present. The notochord is bent at the tip, which will develop into nervous system. Then crescent-shaped semicircular area is present below notochord in the caudal region, and the area of semicircle is well defined. So the dorsal and ventral fin folds are prominent, and caudal rays also they starts in this after 4th day of hatching. Then 6th day of hatching, so it is about 10.5 millimeter in average. Lower jaw it becomes bigger, okay, upturned. Then gape of the mouth, eye is present, notochords are prominent, dorsal fin is present. Then fin buds are seen, air bladder is seen. Anterior part of globular and posterior in the form of elongated triangle is seen. Then dorsal side of the body is yellow in color. Then chromatophores form 2 crescents on the 2 lobes of the caudal fin, which are separated, and they form small colorless area. In the 7th day, lips become thick. Okay, notochord is sharply bent upward, and ventral embryonic fin is folded. Then 8th day, they are sparsely distributed. Okay, dorsal fin and yellow in color. The pigment is near the base of the rays. Annals are present. Ventral embryonic fold results in the ending of the anus. Okay, then dark crescentic areas are present at the base with few black chromatophores, and embryonic fold is present, and pelvic fin along with fin rays are present. Body is golden yellow in color, and dorsal half is predominantly developed, and prominent black chromatophores are present in the head, and black chromatophores are scattered all over the body to form certain pattern. And 12th day of after hatching, so it is 19 millimeter in length. Then maxillary barbels are present. Body become golden color now. Okay, on the dorsal side, and dirty yellow color ventral side. And anal fin is present within 7 days, except at the anterior one, and the rest are branched. And caudal fin rays are 34, and a broad triangular black band on the caudal pedicle are present across the length and the apex facing the head. Then on the 15th day after hatching, so we have anal fin with the 2 to 5 rays, caudal rays, and here black chromatophore band is present, and posteriorly the body is prolonged from the center, passing through the origin of the caudal rays and dividing the fin into 2. Then 18th day after hatching, it is now 25 millimeters in length, and the body is dirty yellow color above lateral line and yellowish white below it. Scales are present, however they are not clear, and especially in the abdominal region. Then black chromatophores are present, anterior margin, and ventral fins are present, and the 2 prominent grayish crescents are present. Crescent is nothing but like half moon. Okay, crescent moon shape. Okay, one on each side of the caudal fin and behind the dark triangular area. And on the 20th day after hatching, so it is 26 millimeter in length, body is golden color, scales are prominent and fully covering the body. 2 dark bands are present above, and 1 band is below the lateral line and along the body of the caudal region, and the dorsal fin is with 3 to 13 rays. Caudal rays are 34, and rays are covered with orange pigment dots. The black band along with the entire width of the caudal pedicle is present in the posterior side, which is again irregular shape on the anterior side. And behind this band, there are two faint crescentic areas which are separated by two colorless streaks. Then finally, on the 25th day after hatching, it is 30 millimeter in length. Now this is the final stage where the body is golden colored with coppery reflection from operculum, and barbels are prominent. Upper margin of eye is faintly orange in color. Then dark band is present in the caudal pedicle, which is again prominent. Upper lobe of caudal fin is larger and pointed towards the rounded edge of the smaller lower lobe. So here you can see the image. So how the changes have occurred till 25th day of hatching. Okay, this is about the Rohu. So what you can do here is, so while answering the exam point of view, you can just write about just introduction about that fish. And so what are the important characteristics? It spawns only once in a year, which is short span of time, all these important points. And every stage, every day, you have to write some 3 to 4 important characteristics, okay, which are distinct from the other stage, and you should write in a point-wise manner. And you can also mention in the pictorial representation. That would be a great representation of writing an answer in your exam. Then second one is life history of other cultivated fish. So here there are some important fish which are commonly cultivated: common carp, silver carp, grass carp, and tilapia. Okay, so we will discuss in general. So first is common carp. So here the characteristics are: fertilized eggs are very small, they are spherical in nature, which is round. Then they are adhesive, whereas Rohu, they are non-adhesive, right? So they are measuring from 1 to 2 millimeters, and the space surrounding the egg is narrow, and the yolk is yellow in color and sometimes light brown also. And embryos hatch out within 2 days to 7 days, depending on the temperature of the water where they are hatching. And the newly hatched larvae, they are 4 to 5.6 millimeter in length, and they have prominent yolk mass. They adhere to the leaves of aquatic plants and by means of cement glands. Okay, and until yolk is partially absorbed, so they remain there. Once the yolk is completely absorbed, which is within 2 to 6 days, depending on the water temperature, this larvae, by that time, they will be measuring 6 to 7 millimeter in length, and they start their normal movement and commence the taking the nutrition. So there is a no need or no use for yolk. So yolk is completely absorbed. So it can actually start taking their own food. So if suitable food is available, the growth is rapid because it takes the food, and fins get differentiated, and within 12 to 15 days, the yolk is completely absorbed and all fins will be developed and they start getting matured. Okay, this is common carp. The silver carp. Then the ova, which is freely viable, which are pale bluish in color, and the fertilized eggs are demersal and round ash gray color. And after 5 minutes of fertilization, eggs start swelling. Okay, next, during the next hour, the process of water hardening is completed. The fully swollen fertilized eggs, now their measure is around 4.2 to 4.76 millimeter in diameter. And the length of the newly hatched larva of silver carp is 5 to 5.5 millimeters. And the yolk sac will be 3.33 millimeters. Height is around 1.25 millimeters. Then height of yolk sac is 1.09 millimeters. Okay. And they have 42 to 44 myotomes. Okay. Which are located behind the, like around 14 to 16 are located behind the anal level. And this yolk sac is completely absorbed on the second day of hatching itself. And now they can start actually feeding from the environment. And the next is grass carp. This fish lays eggs in stages. Okay, eggs are pelagic and undergo development as they float downstream. So these ovarian eggs are of yellow in color and deep golden brown in color. And these fertilized eggs, they start swelling. So within half an hour, they start swelling and they're completely swollen. And newly hatched larvae, they will be around 6 millimeters maximum. Then the yolk mass is present, which is pale brown in color. And out of 45 myotomes, 14 are posterior to the anal level, and 2-day-old post larva, they just after absorption of yolk sac. And the total length will be around 0.99 millimeter in maximum height of the body. Okay. The next is life history of morels. So this fertilized egg is round and non-adhesive. Okay, they won't adhere to, like, they won't attach to any leaves or something. So they'll be buoyant. They'll be floating in the water, free, straw yellow in color, and the yolk is capped by a blastodisc. Blastodisc is nothing but a group of cells, small cells which are divided, and contains oil globule which is liable to disintegration to small globules. So this globule will again disintegrate into small 5 to 6 globules which are unequal in size. And perivitelline space which is present at the top is medium width, and the diameter of egg is 1.2 millimeter. The yolk is 0.9 millimeter. So after 10 hours of fertilization, so this anterior posterior axis is distinguishable. Cephalic portion become broader. Forebrain region is present. Then invasion of yolk is complete. And after 14 hours, so now the egg which have 6 somites, okay, which will again develop into different organs. The somites are they are the cells which are the precursors for the organs when they differentiate. They embedded all over its length in the yolk mass, and optic from the somites only eyes and all this will be formed. So the optic cups are clear, and after 16 hours, mesodermal 9 somites are present, and notochord, which is nervous system, it is discrete, and 4 mid and hind brain regions are defined, and cephalic portion is broadened. And 17 hours after fertilization, the somites, okay, they will be from 13 to 15 in number. Then ectodermal thickening to form lens of the eye indicated. And the tip of the tail is from the yolk in the last somite. And embryonic fin folds appear. Copter's vesicle is visible. And cephalic region is depressed. And after 20 hours after fertilization, somites are now 22 in number. And embryonic fin fold is extended up to 10th somite, and the lens is now fully formed in the eye. Olfactory placode is formed. Copter's vesicle gets diminished. Blood circulation seems to start over the yolk into the rudimentary heart lying anterior to the yolk sac, and the heart starts beating very rapidly, and some dendritic melanophores, they appear over the yolk. Then 22 hours after fertilization, the somites are 24 in number. An embryo encircles the yolk, covers partially the entire capsule, and the tail is free. Olfactory pins and auditory pins are now well pronounced. They are well developed. Some melanophores, like pigment, melanophores, okay, melanin pigment, okay. So the melanophores appear above the neural cord, trunk and caudal regions, and heart beats very rapidly and Kupper's vesicles disappears. And after 24 hours after fertilization, twitching movement of the embryo is vigorous and lashes the tail vigorously against the capsule and thereby springing up the head which suppresses the wall and brings the embryo out. Now the newly hatching larva is dull brown in color and the fin fold originates dorsally in the second myotome, runs ventrally the region of the vent and goes up to the yolk sac, and third and fourth ventricles of the brain are prominent, and 8 dendritic melanophores appear at the caudal region. And these linear melanophores appear in the caudal region on the lateral side of the body. And the next is 3 hours after hatching, the length will become 3.1 millimeter and the height will become 1.4 millimeter, and the heart has 2 chambers. Circulation is around the notochord in addition to brain and yolk. Blood corpuscles are reddish yellow, showing formation of hemoglobin. The anal opening is marked by the slight invagination. So this is how we will see, okay. Then after 8 hours after hatching, now the length is 3.5 millimeter, height is 1.1. Then myotome is 31. Then yolk is reduced. Some more melanophores appear on the ventral side of the notochord and on the dorsal side of the body. And the circulation is present in the optic region. Some pigments are interconnected along the intermyoformal septa. And 15 hours after hatching, the auditory capsule moves closer to the eye and pigments are present over the iris and are now denser. They will form dense pigmentation. And mesenteron is seen as a blind tube and nephroidal duct, which is kidney, like for filtration, are present, and the location of the pectoral fin is marked by thick conical patch. And after 24 hours of hatching, so now 32 myotomes are present, buccal imagination appears, pectoral bud is pronounced, air sac is present, and small tube. Air sac is present, a small tube below the pectoral fin bud, and eyes are fully pigmented. And after 36 hours, now its length is 4.4 millimeters, and pectoral fin is round and membranous flap, and it is used for free movement, and mouth is formed as terminal opening, and vent is formed, and rudimentary gill opening is seen, and nasal pits are differentiated, and thick band of melanophores, they run around the post-orbital region base of the pectoral region. And after 2 days of hatching, the pectoral fin is paddle shaped, okay, and the larvae, they move horizontally in schools, and lower jaw is well developed, trunk region is developed, but it is constricted, okay, only a vestige of the yolk is seen. So here you can see fertilized 2-cell stage, 4-cell stage, 8-cell stage, like that to 24 hours, then hatching, 8-hour-old, 1 day old, 2 days. So finally, so this is how the adult fish look like. Then after 3 days, these pectorals are moving vigorously and they have vascularized, and semicircular vessels are running across, and opercula are membranous, and yolk is completely absorbed. And 6 days after hatching, it is yellow pigmentation is seen, the dorsal side, giving a band-like appearance, like yellow band. And 8 days after hatching, There is a yellow band on the dorsal line with a diamond-shaped broadening at the nape, and laterally there is another band of the same color between two dark lines. Okay, and a broad lemon path occur in the tail ventrally, and rudiments of two fin rays are seen. And the 11 days after hatching, color bands are distinct, and caudal fin rays, okay, they have 5 in number. Hyperals are indicated as basal thickenings. 18 days after hatching, dorsal contour of head is convex, basal thickening appear, and light caudal rays are distinguished. Okay, and the caudal fin is dark along the margin and bright yellow color in the middle, and silver and greenish pigments appear on the orbital rim and also in the post-orbital region. Then 20 days after hatching, the fry assumes nearly the adult characteristics except the color pattern. Okay, and the race starts forming in all fins from the 18th day onward, and there are 14 rays in caudal, 19 in anal, 25 in dorsal, and 17 in the pectoral region at this stage. And the fry is yellow, lemon yellow in color. Okay, and the aerial respiration is noticed. And so the ventral fins appear differentiated or completely distinguished by 23rd day. Okay, and by 26th day they will be almost adults, and it is 18 millimeter in length. The next is life history of cultivated prawns. So generally the eggs hatch and metamorphs, that is it changes into the adult forms. Okay, metamorphosis happens through 4 distinct phases: nauplius, zoa, mysis, and post-larval stage. So here the species, they are highly salinity, they prefer, and post-larval size is 5 to 10 millimeter in length, and they are transparent after post-larval stage, white in color. Okay, expecting for pink collaboration at the tip of the rostrum and the tip of the telson, and anticular pendicle, which is tipped by red, light reddish color. Okay, and light yellowish color on the eye stalk, and a few mid-abdominal segments and telson are present, and a single dorsal tooth is present on the rostrum. The rostrum extends slightly beyond the eye in the first post larvae, and a well-developed supraorbital hepatic spine is present, and a median dorsal and lateral spine is present, and the anal spine is also present. And the post larvae, they swim very fast, moving along the edges and surface, and it is in swimming position, a mid dorsal abdominal bend is characteristic. In the juvenile body, which is young ones, they remain transparent throughout the length. And the red spot is present at the tip of the rostrum, which continues to be present even at the adult stage. On the unropod end, Europod end of telson, a few dark reddish brown spots appear. And the length of the rostrum appears to be 6 to 8 millimeter, and the elongated slender rostrum is the important characteristics for the identification. Then another one is Metapenus monoceros. So it has, so as we said, like different stages, 4 larval stages. So here, so again, each stage is again further subdivided. Nauplius 1, 2, 3, 4, 5. The protozoa 1, 2, 3. Mysis 1, 2, 3, and their characteristics are given. And the late mysis is about, it measures 3 by at the end of the mysis stage, so 3.5 millimeter in length. The rostrum is very small, falling short in the middle of the eye stalk, and the tip of the rostrum is pointed, and the two principal teeth are bore at the base of the rostrum, and one smaller tooth is present in the first front of the principal one. And the dorsal spine is present on the sixth abdominal segment, and post-larval have characteristic brown color. And reddish pigment is present on the ventral side of the abdomen, and in the later post-larvae on the sides, bluish brown spots, they concentrate towards the band. And an M-shaped bluish black patch is also seen on the dorsal side of the abdomen. And the third one is metapenis brevicornis, which have the post-larval stage as 3 to 3.5 millimeter. Rostrum is short. It touches the border of carapace. It is brown in color. Its juvenile rostrum is slightly increase in length. So, while basal elevation is more pronounced and the dorsal teeth are 7 in number, rostral length is 2.2 millimeter, deep yellow in color, and black-red bands are present in the lateroventral aspects of the abdomen, and red spots are on the uropods and tilsons. Okay. So this is about the life history. So no need, there is a lot of information. Okay, so no need of discussing each and every point. So for any life cycle or life history, first you understand what are the stages. Okay, first you remember which day what is happening. Only important points. No need to discuss all the lengths, everything. Okay, only length is enough. All height, weight, everything. Even we cannot remember everything. Okay, so only very distinct points, distinct developmental stages, you have to important points you have to discuss on each stage. And if you have any pictorial representation, you please go through that. You can use color pencil, so whatever the color distinguish, like yellow, brown colors they have discussed, you can use that colors to actually color the figures so that it will be more prominent. Okay, so this is for your exam point of view. Okay, we will continue with the next topic in the next class. Okay, thank you so much.